guys, my name is Abhishek and welcome back to MBBS Treasure Channel. Till now we have completed the general pathophysiology, but now we are going to start a new topic, that is inflammation. So let's begin our today's topic. So friends, now we are going to talk about a topic that is inflammation. In this video I am going to say you about what is inflammation, when this pathological process is actually occurring, and what are the three major methodical processes through which this inflammation is occurring, and what is the etiology or the causing factor of inflammation. And here I have drawn a small diagram to explain the process of inflammation. So this is nothing but a small introduction to inflammation. Okay, so let's start our topic, inflammation. First we are going to see the definition of inflammation. What does the definition says? The definition says that it is a typical pathologic process as a result of injury, and it is characterized by three major processes, that is alteration, exudation and proliferation. So actually what do you mean by that inflammation? Inflammation is nothing but it's a complex response of our body towards the stimuli that causes this injury. And what is the intention of inflammation? The intention of inflammation is to recover the damaged portion of the injury. And how it's going to recover? It's going to recover by eliminating the causing factor of inflammation or the etiology of inflammation, and also by eliminating the necrotic cells or the injured cells. Okay, so inflammation is a good process, but it's a pathological process. It means that this inflammation helps our body, but this process occurs only in condition of pathology. Okay, so inflammation is a complex process that occurs when there will be injury, and this injury will produce the three processes, that is alteration, exudation and proliferation. Now the question comes that what are the etiological factors or etiology or the causing factors of inflammations? Let's see. The etiology of inflammation is divided according to two types, exogenous and endogenous. If the factors that are going to affect the body from outside, it will be exogenous, and from inside, if the factors that will affect the body from inside, and that factor will be endogenous. So what are the exogenous factors? It can be chemicals like toxins, physicals like drugs, then biological, then mechanical. The biological means the microbes, virus, fungi, protozoa, they have a particular type of pattern, that is, what is that known as? They have pathogen associated molecular patterns. Okay, so what do you mean by that? Molecular patterns. It means all the microbes, bacteria, fungi, they have a certain kind of pattern inside them. Like it could be their bacterial cell wall, it could be glycoprotein, it could be polysaccharide. So that pattern will be the exogenous factor or the cause of the inflammations, and it comes under the biological. Then mechanical. What do you mean by mechanical? Mechanical means if there will be any kind of trauma that will be leading to this inflammation, then we are going to categorize that under exogenous. Then the virulent factors. The virulent factors, it helps the biological microbes to colonize in the area of inflammations. Okay, so the virulent factors also acts as a helping hand for the biological or the PAMS molecules. Okay, so these are the factors that comes under this exogenous. And under the endogenous, the complex antibody antigen complex. As we have already read in hypersensitivity reactions, this antibody antigen interaction can also stimulate the process of inflammations. So they will be coming under the endogenous category of etiology. Then blood clotting. As the blood will clot inside and will cause certain kind of changes inside the vessels, and it is endogenous, so we will be classifying under the endogenous category. Then tumors, like what are the tumors? The cancers or overproductions of cells. So they are also going to be the leading factor of endogenous. And then comes the dams. What do you mean by dams? if there will be damage associated molecular patterns. If this is a cell and if the cell will die or the cell membrane will get destructed or the cell membrane will damage, then the dams which were present inside the cell is going to release outside. So these dams molecules are nothing but these are the excitatory protein molecules that are present intracellularly, and they comes out upon the lysis of the cell, that will be stimulating the process of inflammation. Okay, so these are the causing factor or the etiology of inflammation. So we are going to see the process of inflammation when this etiology is going to affect the body. So this etiology can be like the pathogens that we already read, and this stimulus can be the pathogens and the trauma or any kind of toxins, whether it is exogenous or endogenous. Next comes the inflammations. The inflammations occurs in three processes. What are these? It occurs in alteration, exudation and proliferation. And that these three processes we are going to see this in this whole diagram. This diagram shows here, or it says that this is a pin that is going to damage the superficial layer of the skin, and under the skins there are certain kinds of cells. Okay, and the cells are or the tissue are also supplied with the vessels. Now let's see what are the changes that will occur if there will be any kind of pathological condition, and that will be leading to the process of inflammations. So what are the clinical signs or clinical manifestations that we are going to see when there will be a sudden pin will damage the skin? So the first process that occurs is the alteration. What is that? It is the first stage of injury. It is the first stage of injury of structural functional unit of organ. Injury of structural functional unit of organ. This is the skin. Okay, skin is the largest organ of our body. So if there will be a pin that will be affecting or damaging the superficial layer of the skin, then we will be calling it as primary alteration. Okay, this alteration is divided according to two types. It could be primary and secondary. We are going to call it as primary when the injury caused by direct action of the destructive factor or the causing factors. Here the destructive or the causing factor is the pin. Okay, and this whole coming under this mechanical or accidental form of exogenous inflammation. So as this is the destructive factor is going to damage the superficial layer of the skin, then next we are going to call this exact process at the same time the wound that will be made by this pin is nothing but the primary alteration. Then the secondary alteration. What do you mean by secondary alteration? As it says, it's a self-injury which takes place under the action of mediators. So now here comes a certain word. What do you mean by mediators? And when these mediators are being produced, how come these mediators are being produced? Let's see here. This is a pin damaging the superficial surface, and on its surface it contains certain kinds of bacteria. Okay, so these bacteria are going to be released at the site of injury. Okay, so when these bacteria are going to be released beneath the skin or superficial surface, there present certain cells that like macrophage, mast cells, Langerhans cells, dendritic cells, which are primarily responsible for the process of phagocytosis of this bacteria. And as I have said, the bacteria contains these PAMs. What do you mean by PAMs? They may be the lipopolysaccharide, they may be the bacterial cell wall, glycoproteins, any kind of material of bacteria or any kind of material of the pathogens. Okay, so they will be acting as a stimuli. So here the PAMs will be acting as a stimuli. What do you mean by PAMs? It's the pathogen associated molecular patterns. So the PAMs will be released, and when this bacteria is going to affect certain kind of skin cells, then what will happen? The skin cells will get lysed and they will be producing dams. Okay, so they will be producing the damage associated molecular patterns. Okay, so the next process what occurs, the PAMs and DAMs are going to recognize by certain receptors that are present over these phagocytic cells that are present in the tissues. So what are these receptors called? These receptors are called PRRS. The what do you mean by P? The pattern recognition receptors. As the bacterial cell have certain kind of patterns, and in response to them, the receptors are also present in response to catch them or to phagocyte them. Okay, so these are the receptors, nothing but PRRS, pattern recognition receptors, and these are DAMs, damage associated molecular patterns, and pathogen associated molecular patterns. Okay, so after this process, when the PAMs and DAMs are going to attach to the surface of the phagocytic cells like macrophage and mast cells, then the next process is going to occur, the degranulation method or the degranulation process is going to occur. What do you mean by degranulation? The granules that are present inside the mast cells are going to be fused in the surface of the cell membrane, and they are going to produce certain mediators. These mediators are going to be causing certain changes in the area of inflammations. This is the area of inflammation or the site of injury, and these mediators are going to make certain kind of changes at the very place of this injury. So the secondary means the secondary alteration will occur when the mediators are going to cause certain action in the place of site of injury. Then we will call it as self injury or the secondary alteration. So the secondary alteration means the self injury which takes place under the action of mediators. As these mediators are produced by this macrophage or the mast cells, so they are going to produce certain kind of changes, and that will be coming under the secondary alteration. So now let's see the next process that's occurring in the inflammation, that is exudation. What do you mean by exudation? Exudation is the process of escape of fluids, proteins, electrolytes and blood cells from the vascular system into the area of inflammation. Let's see the diagram now. Till now, what happens? The mediators are released. These mediators are nothing but the chemical substances that are produced from the macrophages or the phagocytic cells. What are the functions of mediators? So, what are the mediators actually? Mediators like histamine, serotonin, prostaglandins, these are formed by a complex mechanism reactions, that is arachidonic acid metabolism. As arachidonic acid metabolism occurs due to the damage of cell membrane of the tissue, cells, proteins, so they are going to produce these kinds of mediators in that complex mechanism of arachidonic acid metabolism. We are going to study that arachidonic acid metabolism in a brief manner after this video or in my next video. So let's see, these mediators are produced at this stage. What are these mediators? These are like the histamine, serotonin, leukotrienes, prostaglandins. So these are the mediators that are going to be produced at these very situations. Then next, what will happen? These mediators are going to perform some kinds of actions on the blood vessels. This is nothing but the blood vessels. This is nothing but the blood vessels. This is the anterior portion of the blood vessels, and this is the posterior part. So I have mentioned here that this is the vascular space through which the vessel space through which the blood cells are moving, and these are nothing but the endothelial cells surrounding or making the boundary of the vessel. So what happens? These mediators like histamine, serotonin, prostaglandins, they will act upon these endothelial cells of the vessels, and their functions will be to dilate vasodilation of this region, and they are going to increase the vessel permeability, and they are going to accumulate or they are going to attract more number of leukocytes, more number of phagocytic cells towards the region of the inflammation or towards the region of the site of injury of inflammations. So why they are going to attract this? Because they need to repair the condition of the tissue. Okay, they need to phagocyze the bacteria, dams. So they need extra cell or extracellular mechanisms through which they can totally repair the condition or totally repair the injury made by the etiological factors. Okay, so these mediators are going to perform some actions on these blood vessels, and these actions are nothing but they can be vascular reactions or extravasation of fluid, margination and transmargination, rolling of leukocytes, extravascular reactions like chemotaxis and phagocytosis. What do you mean by this? Let's say for an example, this is a leukocyte, and as we know, or to make you remember, the first leukocyte that to be present at the very first introductions of the inflammation, or the first leukocyte to be present after the process of inflammation, is nothing but the neutrophil. Okay, as we know, the leukocytes are divided according to granulocytes and agranulocytes, and granulocytes, under the granulocytes there are cells like neutrophils, eosinophils, basophils and mast cells. Basophils can be termed as the mast cells, and in case of agranulocytes we know it consists of lymphocytes and monocytes. So they are going to, these mediators are going to attract more number of leukocytic cells, and through which process the leukocytes are going to come? The leukocytes are going to come through margination, transmargination and rolling. The leukocyte will be coming, they will be coming to the margin of the surface, and then through the process of rolling, because the endothelial membrane will be producing some kinds of adherent molecules in the surface of them, so they can pull the moving leukocytes towards them, and through that mechanism we are going to read the margination, we are going to see the process of margination and transmargination. Okay, we are going to see this margination, transmargination and rolling of leukocytes in a brief in a one more video, but in this video I am going to say the overview mechanisms, what is actually occurring in the inflammation. So through this process what will happen, the leukocytes will be, or the protein molecules will be coming to the side or the basal part of the endothelial cells, and they will try to extravasate from the blood vessels. So what is the action of these mediators? These mediators are going to make very big gap in between these cells. Okay, so as they are going to make very big gap in between the cells, then they are going to increase the vascular permeability, due to which the extravasation process of the leukocytes will take place. Okay, so the first cell that will come is the neutrophils, that we are going to see at the site of inflammation. The first leukocyte is the neutrophils. So they are going to extravasate from the vessels. Okay, so they are going to come, they are going to squeeze at this place, and they are going to come to the surface of the inflammation site. Okay, so this very process, or the process of escaping fluid, proteins, electrolytes, blood cells from the vascular system into the area of inflammation, is nothing but exudations. According to different kinds of substance that is going to escape from the bloods, the exudates are divided. That we will be reading when we are going to see the separate video of exudation. Okay, now comes the process of proliferation. See what happens next. When the leukocytes are going to come to this place, the first leukocyte that is coming, neutrophils. Okay, so the neutrophils will act as a vacuum cleaner. What do you mean by that? It will be quickly or in a fast process engulfing the bacterial pathogenic patterns or the substances, and then further it will be producing more amount of chemokines. Chemokines are the chemical mediators through which they are going to perform the process of chemotaxis, means they will be inviting more amount of leukocytes materials, like more amount of phagocytic cells like monocytes, macrophages. So they are going to make the process of chemotaxis, and with response to that, or in accordance with that, they are also going to make the process of phagocytosis. They're going to engulf the pathogens that are present in the region of inflammations. Okay, so next, what will happen as this whole process of inflammations uh, is going to result with a good character. It means they are going to repair the site of inflammation. They are going to repair the site of injury. So the next process that will be occurring is known as proliferation. What do you mean by proliferation? Proliferation means renewing of injured tissue with increasing in the process of cell division. There will be renewal of this whole injured tissue. How the injured tissue will get renewed? Yes, with increase in the process of cell division at this very particular site. After the process of engulfment, after the process of eating the PAMS or DAMS bacterial material, what will happen? So what will happen at the next stage? There will be attraction of platelets or the clotting factors. The platelets or the clotting factors are, or it's going to come in the next stage. And when the platelets or the clotting factors will come, what is their main mechanisms? They will want to clot this area. Okay, so they will bind this wound region. They will attach the nearby tissue in between them, and they will clot the very specific areas. So the function of the clotting or the platelet and the clotting factors is to manage or to clot the wound area. So what is the function of the platelets or clotting factor? Function is they will stop the process of bleeding. They are going to prevent the entry of pathogens inside the into the tissue of the infected area, and thirdly, they are going to produce a framework for repairment. They are going to repair the tissue. Okay, so through this process, the proliferation or the process of increasing on the cell division will occur, and that can occur, this proliferation can occur in two stages. It can be, or it can divide into fibroplasia or regeneration. Fibroplasia means healing with scar, means there will be a present of scar. If the damage will be very severe, if this damage will be very severe, after the healing process, there will be present of a little bit of scar. Okay, due to the presence of this fibrous deposition, because after the platelet or clotting factors will come to this region, then there will be appearance of these fibroblast cells, and these fibroblast cells are going to make the surrounding tissue fibrous if it's a very severe damage. So due to the deposition of the fibrous materials, as they are going to produce more amount of collagen in the nearby region, they will be healed with the presence of a scar, and that healing process with a scar is nothing but fibroplasia. And if the damage is very minor damage or minimum damage, so there will be formation of the healing process without a scar. There will be no fibrous deposition at the site of injury. So that process is known as regeneration. So the stages of proliferation occurs through cell division and synthesis of extracellular components of connective tissue. As as I've said, it could be collagen fibers, it could be elastic fibers like proteoglycans and glycoproteins. Okay, so this is the major important process or processes through which this inflammation is actually occurring. Inflammation occurs through this alteration, exudation and proliferation. So this was all about the introduction of inflammation chapter. In my next video, I am going to say about the actual process or mechanisms through which these mediators are being produced. It is arachidonic acid metabolism. So it will be a very good or a separate video, and it is necessity because it will be helping you with pharmacology, microbiology, and also will be helping with this pathology. So in my next video I am going to say about the mediators, and also I am going to say the clinical manifestations, local and general manifestations of inflammations. Okay, so goodbye, and if you feel this video is helpful for you, please do like, share and subscribe to my channel. Okay, thank you so much for watching my video.